Welcome to a series of conversations with people who help influence our world. Today we are talking to Dr. Subramanyam Vangala, who is CEO of Reagene Pharmaceutical and Biosciences Private Limited. Hello, sir. So, who is also a mentor for people who are in the Bangalore biotech ecosystem. Welcome to the show.
Thank you, Akash, for having me here.
So basically, what we want to understand is right now a lot of people are interested in clinical trials.
Right.
So, for a lay person, what is a clinical trial, and why do they have to be knowledgeable about it?
Yeah, a clinical trial is for drugs. Basically, what you're trying to do is treat a disease with a drug that has a positive effect. May not necessarily for cure, but at least subside the symptoms and makes you feel good. So this is what you need to know is that drugs also have a lot of toxic effects. So you need to find a balance between the safety and the efficacy of the drug to ensure the drug actually has benefit over risk ratio rather than risk over benefit ratio. So that's very important in a drug to be released into the market to ensure that it is safe enough but works very well for the patient.
And going from there, I think that essentially is the process of drug discovery, right? But if you have to narrow it down and probably define it very carefully, how would you define it and how would you expound on it?
So there are two things, the drug discovery is a different ball game. It's a long process. I've seen drugs probably in my lifetime and never made it to the market, or some drugs made it to the market within 7 years, and some of them, and the average time is about 12 years for a drug to come out into the market, because there's a huge regulatory process that we need to be complying with to ensure that the patients are safe. That's the primary concern. Anything can be taken, but if it is not safe enough, you cannot put it on the market. That's the reason you have all these trials that you have. We also start with the preclinical trials, which is called phase 0, and then we go to, once phase 0 shows that animals are safe with very high doses, and then we actually take that data to predict what happens in the clinic. You go with the safest dose possible in the first phase 1 clinical trials. Phase 1 clinical trials are not supposed to be efficacy trials. They're only safety trials to make sure, given long enough, multiple doses, the drug is safe in the humans before you actually do these trials are done in healthy subjects.
So this is toxicology test for healthy patients.
Healthy subjects. I would say I won't call them patients because they don't have any disease. But if it is safe enough in the healthy subjects for a period of time, whatever if the drug is efficacious over 2 weeks or 3 weeks, that period, if it is safe enough, then you immediately go into the launch into the phase 2 trials, which are basically efficacy trials with a limited number of population. You don't have too high population. Then if the drug has some positive effect in the small phase 2 clinical trials, you go into the phase 3 extended trials with a large number of populations. In some diseases, you don't even have 100 patients. But if you take heart disease and diabetes, etc., you have in millions of patients. So these clinical trials are done maybe in 4,000 to 5,000 patients. Just so the clinical trial design and the patient recruitment also depends on the number of patients available to recruit. So in some cases, recruitment itself takes 1 to 1.5 years. The trial may be only a 3 months trial, but recruitment itself can take 1 to 1.5 years. So these are the things that take a long time to be able to do entire drug discovery development program, all the way from drug discovery to the market, to convince the regulatory agencies that you have a very good drug that will treat particular disease, but at the same time it's safe enough to be given into the patients. So this is a long winding process and very costly, and 60% of the budgets go to the clinical trials. Before that, it's only about 30 to 40% you spend. But 90%.
What do you spend the 40% on?
Doing discovery chemistry, medicinal chemistry,
Basic
biology,
research,
pharmacology,
fundamental
to
research.
develop an animal model and see how the drug works. And then you also have to do preclinical safety trials in 2 species at least, in rats, maybe dogs and monkeys. So depending on those data only, you can nominate a compound to the clinical trial. It has to be safe enough at least in 2 species. So if I take rat and dog, and it is very toxic in dog but safe in rat, then I may not be able to take it to the clinical trial unless I show 2 species. So I also sometimes recruit monkey studies to be able to show that it is at least safe in rat and monkeys, not in dogs. Dog is not an ideal species for the human relevant species. So we actually, if the monkeys also trials are very good and safe, then we take it to the clinical trials. So this decision making process is long and winding. Sometimes
How long are we talking here?
some drugs take 15 years to market.
15
The
years,
fastest
what is the starting point of 15 years? So from the time you have the molecule concept,
Yeah, from the concept I'm talking about. So I have one target and say, hey, this is a very novel target. Nobody explored it before. And I want to take this, see if I have a drug that affects this target and make sure that that particular disease, maybe cancer or diabetes or whatever the disease endpoint you're looking at, and does it help in that disease. So the target is very essential in selection. And then what you do is you design drugs around the target. So the medicinal chemists come in and they actually design to see whether the chemistry is optimal to synthesize chemicals that will modify the target to have a positive effect. Once that is done, then you take into the animal models, and if the animal models show positive effect, then you go into the preclinical safety trials, animal trials. These are regulatory. FDA, ICH guidelines, EMA, they have several guidelines on how do you do these trials, and these are all protocol-driven studies, and make sure that you do them by the book.
By the book.
Yeah.
And these are essentially regulatory, civil regulatory agencies,
Right.
which kind of put
Right.
the guidelines.
Right. So if you think you have to modify these protocols and customize to your needs, you have to get pre-IND meeting with FDA to or regulatory agencies to make sure that if they agree, regulatory agencies agree. Oh, okay, he cannot use monkey or dog. He needs to want to use hamster or some guinea pig or something else. We need to get a regulatory blessing on that to make
Okay.
sure that before you do those animal trials as well. So these are the things that we need to be able to convince the regulatory agencies that we do by the book, because if regulatory agencies do require safe drug into the human. That's their primary objective.
Yeah, protect
Right.
consumers from the harmful effects if
Right.
they
So
may.
I'll give you one example here. You've seen the saga with hydroxychloroquine in the last
Correct.
10 months.
It's been in the news.
Yeah, it's been in the news for the COVID treatment. And the last 10 months, you've been seeing a lot of doctors and clinical trials are done. And everybody says that hydroxychloroquine is a good drug. It has some positive effects on the COVID disease. But FDA didn't agree to it. And FDA first gave emergency use authorization. After a month later, they revoked it. The reason they said was that because most of the people who are taking hydroxychloroquine also have comorbidities with other the disease. Those drugs may interfere with this drug and have severe side effects. The primary one is arrhythmias.
So hydroxychloroquine has comorbid effects with arrhythmia patients. Meaning
Not
like what?
arrhythmia patients. I mean, if I'm diabetic, if I'm taking metformin, just giving an example.
Metformin, yeah. Okay.
Taking metformin, and if metformin and hydroxychloroquine interact with each other, the hydroxychloroquine or even metformin go to higher level drug concentrations that may impact your heartbeat.
Okay. So these
These are the reasons why they say that. Another reason I actually wrote a review article on this. that just published last 2 weeks ago in Pharma
Okay.
Bio, just showing that if you give a single dose of hydroxychloroquine, half-life is 22 days. I mean, it lasts forever. So if you want to reach a steady state by giving daily doses to hydroxychloroquine to humans, it will take 6 months to get a steady state concentrations. So, which means that it is accumulating in your body.
Okay.
And somebody else already suggested with the lupus patients is that you
Lupus
don't give
Pharma,
daily,
yeah.
you don't give daily doses. You give a dose once a week.
Okay.
That way you don't have this accumulation problem and you cover for the safety. And these are the things that doctors also should know to ensure that they shouldn't be giving the drug every day. They should be giving based on the pharmacokinetic parameters like half-life, volume of distribution, other things. So these are the things that determine how efficacious or how safe the drug is in the patients.
And you talked about interaction effects of drugs.
Right.
So what about hydroxychloroquine? Consumption of hydroxychloroquine without any interaction effects.
So hydroxychloroquine, in my article I also wrote that there are quite a few drugs interact with hydroxychloroquine. Many times hydroxychloroquine actually inhibits those drugs' metabolism or some other part, and their drug concentrations go up. And they are the ones who are causing. For example, azithromycin is there. Azithromycin is very well known a cardiotoxic drug. Initial trials with hydroxychloroquine were done with azithromycin.
Okay.
And this is where everybody said it's working very well. Azithromycin is just an antibiotic.
Okay.
It's not an antiviral drug. Hydroxychloroquine is also not an antiviral drug, but it seems to have immunomodulatory effects, whereby it may inhibit the cytokine storm. It's a passive effect, but it doesn't directly inhibit the virus or kill the virus, hydroxychloroquine. So both of them together have side effects that are really life-threatening. And this is where FDA came in and said, hey, we have all these clinical trials done. I think I've seen 24 or more clinical trials on the hydroxychloroquine itself. But the FDA final conclusion is that hydroxychloroquine is not safe enough to be given to the patients. Please do not use it.
Okay. That's why they, removed the emergency authorization
Right,
that
exactly,
they gave.
yeah.
So right now, are they? Is it part of the therapeutics life, cycle? Meaning, like, are they prescribing that for patients who have this COVID disease?
So for the COVID disease, I mean, there are lots of drugs out there. Our own Biocon, Torozumab is there, and many other cytokine inhibitors. These are all the drugs that these protein antibody drugs actually inhibit some of the cytokines that are elevated during the viral infection. Any viral infection has inflammation associated with it.
Inflammation.
So they stop the inflammation or reduce the inflammatory response to be able to, for the patient to be able to feel free. Because the inflammatory cytokines also have harmful side effects.
Okay.
And that actually is a double whammy for a virus because it's actually increasing the cytokine storm, which is supposed to kill the virus or stop the virus from getting infecting more and more. But these things come and kill your own cells, basically.
Okay.
Cytokines.
Okay.
So they don't discriminate between the virus and your normal cell. And that's when the inflammation actually becomes compounded effect and the patient goes into a much more serious condition.
So, and the thing that we have to understand, I think there was one term that we initially referred to but did not quite define for the layperson. There is usually a target for a drug, before you conceptualize something in the lab. There is a target. So, what do you mean by target for the layperson to understand?
In fact, traditional drug discovery back until 1980s and 1990s is called a phenotypic approach. Phenotypic approach means you know symptoms of the disease.
Correct.
And you give a drug and see if those symptoms disappear or subside. So basically that's how you call it phenotypic. But the problem with symptoms is that 10 diseases have the same symptoms. So you don't know which disease you're controlling because you may not be treating the right disease symptoms. So the problem is diagnosis is very important, right? So
Correct.
for diagnostics, there's some genetic defect or a protein defect or something else biological defect is there. Identifying that biological defect is called target identification.
Okay. And this is in the body of the patient.
Yeah, in a patient or
It's a population.
disease, yeah. And that target is the crucial one that you're targeting and say that, okay, inhibiting this target or activating this target, either way can subside the disease based on the biochemistry and molecular biology of the disease process. And what you're saying that, okay, I have an effect on this particular target on a outside, you take the target, put it in a test tube, and test your drugs and see how well they bind or inhibit or functional assays also in cells. And then you come to a level that my drug is actually having positive effect on this
On this
target. And then what you do is you want to design more drugs around it to make sure that you have a more positive effects than side effects because you're looking at the biochemical parameters and other modulators in a cell systems, unicellular systems where this target is overexpressed. And then you say that, okay, there are some biomarkers, we call them. If the target is modulated, it creates some biochemical effects around it. So those biochemical effects spill out some biomarkers, we call it, either protein biomarker or a RNA marker or something. And you can use that biomarker to say that my disease is getting modulated. It may not be treated, but at least my drug is having an effect on this target. It gets more complicated when you go to the clinical trial. These are easy to do in cell systems, but in patients you cannot do so easily. So when you go to the patients, you don't even know where the drug is going in your body. So there are lots of innovative clinical trials, very expensive models are there that pharma companies are employing in the US right now to use the microdosing technology where it is the lowest safe dose possible that you administer to the patient and see there are some labels that you radioactive labels, specialized radioactive labels. You put it on the drug and see if the drug actually reaches the target by actually imaging analysis.
Okay, so the idea is you take the drug, you isolate the target, you check the effect of the isolated target afterwards through imaging.
Right. So what you do? What you do is you're actually imaging the patient.
You're making the patient.
Imaging the patient.
Oh, imaging the patient. Okay, got it.
Right.
Yeah.
You're.
I mean, how often? I mean, you take the drug today. You take the image the next day, or how often do you have to image?
Yeah, it depends on the label that you're putting on the drug. That labels, many of these labels that we use on these drugs are very, very short-lived.
Okay.
So you have to do it immediately or.
As soon as somebody takes the drug, you just put them in an
right, and see where it is going in the body. That's it. Where it is going in the body. You want it to treat Alzheimer's disease, it has to go to the brain in specific locations. And image brain and see if that drug is there or not with the label through an imaging process.
We'll get to Alzheimer's if we can. So I think one of the questions that had written down was, aren't there enough drugs? Which seems like a tautology in COVID world because COVID is a nice scenario which shows the world that there aren't enough drugs. So that claim is sort of self-explanatory now these days.
Right.
What do you have to say on that?
Yeah, I mean, see, to go into the disease, apart from our regular diseases like diabetes, and we understood very well diabetes, maybe atherosclerosis, we understand very well. We have enough drugs around. Antibiotics, we know, we've got tons of them and everything. The problem comes to is that rare genetic diseases. There are more than 6,000 rare genetic diseases in humans.
Okay.
This is where the available drugs today are not sufficient to be able to treat all these variable 6,000 rare. For example, you take autism in the kids, right?
Okay.
I read about a few years ago that autism is not a single monogenic disorder. It's not a single gene disorder. There are 800 different mutations were detected in different autisms.
So there are 800 genes, they all interact and they all express or suppress themselves, and
There are mutations, point mutations.
okay.
800
Changes, yeah.
different mutations. The same gene could have 10 different mutations, or
Yeah.
10 isolated. You may have one mutation, I may have another mutation. Like that, there are 800 mutations were detected that predispose these kids to autism disease.
So what is the pathway for a challenge as
So
complicated as
this
this?
puts a lot of pressure on the research R&D programs. Because now you're not looking at a all or none effect, right? So what you're taking, you're not saying that all these 100 patients are the same anymore.
Yeah.
Each individual patient may, out of 800 mutations in 100,000 population of people, you have to divide and say, hey, 20 people may have this mutation and 20 other people. So you have to devise a drug for each 20 patients. You cannot have one drug to treat all these 10,000, 100,000.
So essentially personalize it as much as possible. And
Absolutely.
The
That is
sample
where
size,
we are coming to. Yeah.
and the sample size becomes smaller and smaller because the recruitment of a very specified targeted group is hard because they're all spread out everywhere.
Right.
If they're spread out everywhere, the cost of recruiting is higher.
I'll give you one, just nothing to do with drugs. I'll give you one example. Cigarette smoke. We know it causes lung cancer.
Yes.
You know how many people actually get lung cancer?
How
Less
many?
than 20% people who smoke cigarettes.
Okay, even amongst the people who smoke cigarettes.
Smokes, it's chain smokers, maybe 2 packets a day. I've known a lot of friends who've been doing that for the last 40 years. They don't have any symptoms of cancer or lung cancer. But the only 20% of people get cancer. What is the biology behind them getting that cancer, lung cancer, as opposed to those people who don't get cancer? This is the trickiest part because, out of those 20% people also, there are subgroups again. Some people get interstitial lung cancers, there's some mesotheliomas. So the cancers are also different in this field. So that makes it harder to nail it down to what is the biological effect of a drug or anything that you're trying to treat these cancers.
If we thought the thought of drug discovery was tough enough, looking at the COVID situation, the picture that you're painting with quite a broad brush seems to scare anybody who even thinks about this, because the R&D challenges given this scenario can be extremely difficult in terms of how you recruit patients, how you recruit people for clinical trial, how do you ensure that your recruitment processes are such that, obviously you randomize it enough,
Right.
but you have to still look for people who would be conducive for the study.
Right.
And it becomes very hard given your scenario.
So there's a disease called a Pompe disease. It's a glycogen storage disorder disease. So
Okay.
kids who are not born with that disease, most of them die by age 15. Some survive, but it's called adult Pompe disease. But anyway, the science from a lot of other companies in the US and everything, what they did was they cloned the gene for this enzyme that is deficient in these patients. And what they did was they expressed it in vitro in other cellular models, mammalian cell models, and synthesized this protein. And then they put it in the bottle. The patient can get injections.
Okay.
Okay. In India, I looked at how many patients are really there. So far, I think 20 to 25 patients. Now, that American drug, to come and give it, I think the cost is more than $100,000 per year. Here, I think so.
100,000 a year. That's
I think so, if I remember correctly. I got to go back and look at the numbers, but it's a very expensive drug. That families are putting their entire
life earnings.
livelihood onto treating their children.
Yeah.
Okay, so that expensive. In India, importing the drug and treating these patients it's impossible. And this is where the science has to discover more mechanisms on how to treat these diseases cheaper way rather than doing, and we are trying to do that. We've got some ideas on that and how we can develop a smaller molecule drug repurposing. There are drugs out there in the market, as I said, there may be 3,000 to 4,000 drugs are already on the market. One of them should be able to work for these glycogen storage disorders. We are looking into it. Hopefully we'll be successful coming out with a drug that may treat with a 10 rupees rather than 10 rupees a day. You give it a lifetime for a patient, that's nothing compared to $100,000 drug to treat a patient. So these are the things that to be innovated to be able to reach out to the patients that really need these drugs.
This will be a nice time for a plug for the company, right? So Reagene Pharmaceutical and Biosciences. So what does Reagene mean?
So there are different angles to it. In fact, we conceptualized this idea back in 2006.
Okay.
Myself and two other friends. The other friend who lives in Hyderabad, he was a former CSO, Dr. Uday Saxena of Reddy Labs, and he, myself, and another gentleman who's a serial entrepreneur. He just filed a company for IPO in the US, and we actually conceptualized this idea. We were actually roommates in Canada while doing PhD.
Okay.
So
all three
of us. We were all roommates in the same room, and there was another physics graduate student who was there, but he's not involved in biochemistry, life sciences research area. But now we come together after nearly 40 years, and we said, what can we do with our experiences put together?
Hence, the moral of the story is treat your roommates carefully because they will become your partners. So, and what does Reagene do?
So the conceptual, we three actually met in 2006 sometime in New Jersey at my home, in fact. And at that time we conceptualized an idea, just sitting there at the dinner table, and we conceptualized it. We had a couple of meetings, but we couldn't be worked out because we all have daytime jobs. Full-time daytime jobs, right?
At the time you were working with,
conceptualizing this idea.
and you were with J&J at the time?
I was at J&J at the time. Satish was an entrepreneur, and at the time he was working for a company called Nucleonics, his own company.
Okay.
And Uday was full-time with Reddy Labs, who was in Atlanta, Georgia, built their drug discovery programs in the US. So we all had our own daytime jobs. None of us were able to diverse our jobs, right?
Correct.
And that's pushed the timelines. And in 2016, I approached the Saxena again. Let's start it.
And the one-line idea at that point was?
The idea was to create reagents and based on the need. For example, in India we import a lot of reagents, biochemical reagents. Right, they're all expensive. If I have to buy something, it will take 3 weeks to come, and I have to pay customs duties and also other fish shipping charges and everything. In fact, in the US, I used to get the same reagent next day on my table when I ordered it.
Okay.
Here, I forget the idea by the time that reagent reaches to my.
So you forget the idea for which you ordered the reagent.
This is the problem. One of the things that we need to solve the problem in India. In Indian context. So we're building reagent database in India, make in India for India reagents. That's for the primary concept. Okay. Now but we are not looking at reagents as such biochemical reagents that are available in Sigma or anything else. We are into specialty reagents solutions company. So what we are trying to do is, right now, our company has evolved into that concept stage into 3D bioprinting tool reagents.
3D bioprinting tool reagents. Okay.
So they have different applications. One is for drug discovery, one is for toxicology, one is for, patient trials. They have 3 different models. Our idea is to be able to develop these proprietary tools and patent them and license them out. Okay? If you all want tomorrow that you want to use my tool, pay me and I'll give you the tool. And you can do your own research in your own lab with that tool. Whatever the way you want it, customized to your needs. Or if you want to outsource it to me and do it, I'm not big proponent of SC as a CRO, but I would like to see if there's any collaborative programs between us that we could actually benefit together.
So
Either a revenue sharing model or something else we could think about it.
So the end product is a scheme or a reagent?
So a tool. It's basically a tool that is we conceptualized it and brought it to that market. And that tool can be very useful for a pharmaceutical company. It can be very useful for a CRO company, another CRO company. It can be very useful for a toxicology company, industrial toxicology company. All of these can use this tool. Okay. And the tool may be 100% perfect, but we can fine tune it for your need.
So when you say a tool, you're talking about a software tool?
This is not software. We are also going that way. So what we want to do is actually, this is right now we're doing it in terms of collaborating with Tech Mahindra, and especially on the COVID program we're doing it.
I think you announced it a few months back.
Right.
Yeah.
So we are also looking at bringing the artificial intelligence tools.
Okay.
And combine with our 3D tools. As I said, 90% of the clinical trials fail in the humans.
90% of the clinical trials fail in the humans.
So as I said also that 60% of budgets are spent on the clinical trials. So if I fail in phase 3 trial, imagine I already spent close to $1.5 billion.
$1.5 billion with a B, meaning close to 10,000 crores.
Right. So you put them into the drainage. That's it. You got to the drain. If your phase 3 trial fails. So the question is this is the question we asked. How do you improve the success rates of the clinical trials? I said 90% fail. Can you bring them to 50% failures? Means 50% actually success rates.
And the way you think you can do that
is because animals are not capturing the human physiology. Okay. So animal models that are being done, these are regulatory mandated studies. Ours is not a regulatory tool yet. We plan to make it a regulatory tool one day, if possible. But it's a right time to do it because there are also a lot of animal ethics issues are also involved. Just killing the animals for no drug is coming out is also an ethical issue, basically.
Yeah, correct.
Right?
Yeah.
So, for that reason, if we can bring a tool and show to the regulatory agencies that our tool is better than animal tools that can capture. We're not saying to regulatory agencies that get rid of the animal trials. Maybe you can prioritize our trials first and only do the animal trials that are minimum required. But EPA, Environmental Protection Agency in the US, has already promised by 2035 they'll completely eliminate animal studies by using 3D bioprinting tools.
They have said that?
They've said that. They already gave a public
announcement. So animal testing is completely gone by 2035?
Exactly. They promised it. So this is kind of research that we need, but what is missing in this toolbox is the reagents that predict the human outcomes.
Okay.
What I mean by reagents is that if you take a liver, right, human liver, liver has 5 different cell types.
Yeah.
They all interact with each other in the liver.
Okay.
If I put one drug and put up one enzyme there, the cascade goes on, you don't know what's happening in the liver. Liver. That you cannot study with an animal liver.
Understood. You understood. So
how do we get it? So there are different ways of creating these organoids. People are doing it now. What we are doing is a 3D bioprinting. Organoids, spheroids, 3D bioprinting tools. What people are doing is they're combining these 5 different cell types and creating an organoid. Or they're differentiating stem cells to liver-like organs. Okay. So there are different ways of doing this, but everybody's idea is to be able to create human-like liver.
Human-like liver
that I can work in outside.
In the lab.
In the lab.
Yeah. And how many companies are there which are doing something on those lines?
In the US, there are lots of companies. I can't even count US and Europe. There's one company called Emulate that has made miles. Right now they're even working with FDA to actually make this model toxicology relevant model for human development. Because as I said, many drugs fail in the clinic due to safety issues. Primarily is a liver safety issue. The secondary is the kidney issue. Not kidney, I mean heart issue, and kidney issue third one. So they're primarily creating for that reason, can they reduce or minimize the liver toxicity failures by using this liver organ on a chip model, and they're collaborating with the FDA to be able to screen a lot of compounds. You know, you and me only can look at the literature on the PubMed and everything that we know what's published. A lot of companies don't publish their data outside. That data is actually with FDA.
Correct.
Right? So if you collaborate with FDA, FDA is going to give you the drugs that you don't even know that exist and died because of the hepatic failures.
Meaning like there are some private markets that, you know, these are private companies which are submitting to the FDA, so they are
right, exactly. But they don't many times give the reason why their drugs fail.
Yeah.
That information is only with the FDA available.
Okay. So collaborating with FDA is one way by which you can mitigate your prospective.
Yeah, because the FDA knows more than what we know outside in the literature.
Yeah, understood. So just to understand, take a step back and understand your personal journey. Like you went to Canada for your graduate school, right? So
right.
The motivation to go to Canada was obviously at the time. This is
1981. I left.
1981. So at that time, US was like a land of opportunity. Let's say North American continent was like a land of opportunities, and people who were pursuing science, specifically let's say biology or chemistry, were going there in droves. But you were part of that whole, let's say, group of people, or like you thought like this was an interesting thing for me personally?
Okay, I think I have to go back to 1979.
1979. Okay.
1979, I actually finished my MSc in biochemistry in Andhra University.
Okay.
I was clueless, I tell you, like anybody else at that time who graduated with an MSc degree, I was clueless.
Okay.
Don't know what next. But the only thing I know that I want to stay in biochemistry. That's the only thing I knew. Many of my seniors and others, they went into IPS, IAS, IFS, and all the civil services, and also bank probationary jobs and everything, because there were no jobs in biochemistry.
Back in 80s,
There were no jobs. There were absolutely no jobs at the time in India. You get degree, but don't know what to do with it. I came to IISc here in Bangalore for an interview. I was not selected.
Okay.
I mean, it's not me, but they have their own way of selecting people. And I was not worried about it. I went back and I went to IIT Delhi in the biochemical engineering department for a PhD interview. I'm not an engineering guy, I'm a biochemist. So they asked me some engineering questions I could not fare well, so I could not get there either.
Okay.
Then I saw an advertisement for a senior scientist job, some scientist job in CFTRMI, so I applied for it. My classmate got it. Now he just retired last year, but I didn't get it. So these are all the things that happened to me. I finally took UGC scholarship in Andhra University and stayed there. But my ambition was telling me that I need to go somewhere. So I was walking on the road, and my old friend, who was just leaving to Canada to do a PhD, but he's a statistician. He got his bachelor's degree in ISI Calcutta and MStat in Delhi. And he said, I got the Waterloo PhD program. So that prompted me to actually go and look for universities in Canada and the US in the library. I used to spend 4 to 5 hours in the library and getting their applications and applying in the US. I didn't have GRE at the time. Okay.
Was
so,
that a mandatory thing for applying to GRE?
For many universities, it was a mandatory thing.
Okay.
But again, some people were giving me admissions, but no scholarship, no nothing. If you have your own money, you can come over.
Okay.
But my father was a professor in the university. We were not I could not afford to go on my own. So I kept writing, right? And there was one university finally in Canada, Newfoundland, St. John's. Suddenly a professor writes to me, Peter J. O'Brien. I mean, that's why sometimes...
Which university is it? I didn't catch it.
Memorial University, St. John's, Newfoundland, Canada.
Okay.
That professor wrote to me saying that I saw your application, you want to do cancer research. My lab is doing cancer research. If you're interested, I'll submit your application to the Dean of Graduate Studies and get you $6,000 scholarship. On top of it, you can do teaching assistantships and I can also pay you some from my own research grants.
So you got a complete scholarship.
Scholarship about $10,000.
Okay.
My ticket to Canada. That was it. I never had to look back.
Correct. So when you land there, then you meet Uday Saxena, your co-founder.
No, not immediately. What happened was I was working already at Peter O'Brien's lab, and this was January 1981.
Okay.
And he comes there at May 1981.
Okay.
That's when he worked for a neighboring lab, Professor Mukherjee, who was the head of the department of biochemistry at the time. So that's how we became friends. Satish Chandran, as I already described to you, was working in the molecular biology department in School of Medicine. And
okay.
He also joined in May 1981. We became roommates.
Okay.
Long-term friends for nearly 40 years.
Yeah.
Okay.
So you finish your graduate school and then you go
So I got a postdoc offer in Toronto, University of Toronto.
Okay.
But I looked at it, even Canada doesn't have many jobs, to tell you the truth.
Okay.
I mean, this is reality. I mean,
Yeah,
you got to do the reality check. You got to.
What is the population of Canada in 1988?
Maybe at that time it was 22 million. I think it's about 38 million or something.
Now, yeah.
But there were no jobs. I mean, tell you the truth, I could become a postdoc for life there. And I've seen a lot of people just stayed as postdocs in Canada. And I said, there's not many industry there. Pharma industry was really non-existent there.
Yeah.
So I could live my life as a postdoc, and if I become lucky, I can become a professor there in the university. There are not that many universities, about 20 universities.
Yeah.
So luckily, one professor from Colorado who read my publications, a very nice person, he also called me up and said, hey, I read your publications, I have a postdoc position, I advertised in Science. There are 75 applications, but you didn't apply for it. I want you to come and join my lab.
So you did not apply. The professor called you and asked you to apply through this.
And he took me for an interview, and then right away he offered me a fellowship.
That was so nice of him.
Right.
Yeah.
But sometimes life is luck. I'm telling you, you have to be at the right place at the right time. And he gave me a 2-year postdoc position. Okay. I don't know what I'm going to do at the end of 2 years, right?
Yeah.
I went to a conference. I was presenting a poster at toxicology conference. There was this gentleman comes to me and says that, are you Manny? They called me Manny at the time. In Newfoundland, they used to call me Superman. That's another story that I will tell you later. But that guy, he's a full professor. He's not even 30 years old at Berkeley.
Okay.
Not even 30 years old. He's got 350 publications on his track record already at that age.
Okay.
And he approaches me and he says that I was ready to explain my poster to him, right? He was not interested in listening to my poster. And he started talking to me. So when is your postdoc going to finish with David Ross in Colorado? Maybe end of December. We're looking for recruiting people on a non-tenure track level right now. Like a research assistant professor, not funded by soft money, but in 3 to 4 years, if you can get a grant of your own, you can become a full-time assistant professor in Berkeley. But I want you to think about it. I'll recommend to the head of the School of Public Health, and we'll recruit you into the School of Public Health.
School of Public Health, okay.
University of California.
University of California, Berkeley.
Then I just went there and they sponsored for my green card and everything I got. I wrote 2 to 3 grants and none of them got funded, unfortunately. So the industry started knocking on my door.
And how long was it? How long was your tenure at Berkeley before you thought
I joined in 89 January in Berkeley. I left 92 September.
Okay. So in 3
Little
years.
close to 4 years, little
Okay.
close to, yeah. By end of the year it would have been 4 years, but yeah. But anyway, industry started knocking on my door.
What industry? I mean, obviously it's pharma, but which was the company?
Pharma, American Cyanamide, it doesn't exist anymore.
Okay.
You heard of this gentleman that discovered antibiotics, anti-cancer drugs, very popular Indian guy. I forget the name right now.
It's okay. We'll put it up anyway.
Right.
So you got but you were leaving Berkeley for a private job. That also means like you're giving up on your, let's say, academic
interest.
aspirations. Right?
It was a difficult decision to make. I was looking at benefits and risks of leaving the Berkeley job. I may never get a grant of my own if I stayed back in Berkeley.
That's also
right?
true. Yeah.
That's also, I need to weigh it out.
Yeah.
Whereas industry is offering twice as much salary,
Yeah.
and they're also giving a lot of stock options, bonuses. So I had to make a decision, and where do I want to be? Opportunity came to the door. If I leave it, I don't know when I'll get another opportunity to do it. So I took a quick decision in 1992 September. I moved to American Cyanamide as a senior scientist.
Okay.
And luck again, there was another gentleman who I was not reporting to, asked me to solve some clinical problems for a compound. It's in the clinical hold.
Okay.
So with my biochemistry, molecular biology knowledge, I solved the problem, and FDA withdrew the clinical hold, and they went back into the clinical program.
Okay.
So this gentleman, I met him in 1992 December, and I worked with him till 2007 sometime. Okay, it's almost 15 years I worked with him. He moved to Purdue Pharma. I moved with him as a group leader, and he moved as a global vice president to J&J. I moved him as a site director at J&J in New Jersey.
So you were his protege, so to speak.
Absolutely. I mean, I think I'm lucky to be associated with people who like to work with me, and I would like to work with them. It's a kind of chemistry we had together.
Exactly.
And which benefited me a lot and benefited them too. It's a mutual beneficial. We worked good together basically for 15 years, me and Dr. Alfred Tonelli.
So this entire tenure ensured that until 2006 is when you thought like you should start a company of your own. So what made you think that? Where the opportunities at company as big as Johnson & Johnson were not ripe for innovation inside the company?
Yeah, so Johnson & Johnson was also trying to move a lot of operations to China. Many companies were doing it at that time because China is cheaper, and also there's a lot of money going into China. So as anybody else, even J&J was thinking about it, and in fact, in front of me there were about 500 medicinal chemists and biologists were laid off on one day. And I realized that when J&J is not going to stay on the same course of drug discovery for a long time, they may move their operations elsewhere. So at that time I decided to move out, and I moved to Shire Human Genetic Therapies. But at that time I got calls to move into India to build infrastructures. One company picked me up with Sai Life Sciences right now. It is called Advantium one time. They picked me up to build their infrastructure for biology, pharmacology, toxicology, biochemical, primarily DMPK. Built their infrastructures for it. So then I moved on, came to Advinus, and they already have the infrastructure to nurture the people and bring them to the speed of what's happening in the industry. So I actually managed the teams at Advinus also. So I built J&J's ground up DMPK Advantium steam here, ground up.
Yeah.
Brought them to a level that it's self-sustainable. But I have to move on in my life.
So these tenures were essentially training wheels for you to start your own company.
Absolutely. I got confidence at one level. And in fact, in 2012, I was actually thinking of starting something. My better half knows me more than anybody else. And she said, you don't have business acumen. Do you think you can do it?
Okay.
That's very important. You need somebody to tell you what's the weakness in you.
Correct.
Right? And who can better than your better half.
Yeah, exactly.
Right?
Did you talk to Uday at the time?
At that time, I didn't talk to Uday. Was still in Atlanta. Was working on second generation company with Dr. Reddy's. So he was still tied up with his own commitments. So in 2016, when I talked to him, when I came out of Advinus, he was ready. So that's how long it took. Because I didn't want to start on my own. Because you need to have partners who are also like-minded and who can tell you what you're doing, what you're doing right and what you're doing wrong. You need those kind of minds along with you. This is kind of a constructive criticism as well as appraisal.
Exactly.
You need to be aware of it. Don't take anything criticism personally, and make sure that there's something right in this, or I can take that advice and make sure that I don't make that mistake. So if somebody can analyze themselves, what is the self-awareness basically is. It's very important in life. And if you don't do the self-awareness and you think you know it all, you'll never succeed in life.
You can go off the course, and before you know, you're just about to drop from a cliff.
Right. Yeah.
And there's nobody to stop you. And worst of all situations, you're accelerating towards that end of the cliff.
Right.
So it might happen. So it's a good idea to always have partners
who will give you the best advice that you can take, and that's how you have to live it. And in life, you take advice from people and see what is good for you, what is not good for you, and make informed decisions.
Were there any cultural shocks from, let's say, India to Canada, Canada to US, and let's say, vice versa?
Yeah, initially you'll have culture shocks, but I do believe the phrase that some people say that the saying that be a Roman when you're in Rome. So you have to adapt to new cultures. I think I've done a good job of adapting. Wherever I go, I adapt to the conditions. This is very interesting. In 2007, that Alfred Tonelli, who was my prodigy, I told him I'm planning to go back to India. There's this Indian guys who want to start a toxicology company, want to bet on me about 40 to 50 crores to start a company in India. What do you think about it? He gave his honest opinion to me. It's not that I don't want you to go back to India, but I'm asking you, how long have you lived in the US and Canada together? 28 years to date, I said to him. So what do you know about India? I said, I go once in 2 years for vacation for 1 month, but does it tell you how the business runs in India? So, you know what? Yeah. My honest opinion is that don't take this offer because you'll fail miserably.
Okay.
It was honest opinion. It was honest opinion. If you want to go still, take a job first. Learn the know-hows in India.
Okay. Don't bet on your own money.
Money right now. Right. I was not bidding on my own, but somebody else is bidding on me. But again, I think these are the things that you need to take it. You have to analyze it and say, yeah, there is a truth in this. They're not criticizing you for not becoming an entrepreneur, and you have to realize it and make sure. And that was a good idea he gave me because I came to India took a job inside Advantium. They built their infrastructures. I met a lot of people, and I understood India is different from when I left in 1980.
How is it different, and what are the key, let's say, pointers that someone who was in the same situation as you, who wants to, let's say, move back and either jump into entrepreneurship because he is a subject matter expert, or for that matter, he just wants to move back because it's a better cultural fit?
Uh,
What are the key things that you have to remember?
I don't know if it is advice or just my viewpoints are that, I mean, I would love Indians to come back to India. I mean, I came back, my edition came back. I never regretted it. Okay. And the same thing is that you come here and keep complaining about India and this and that, you'll never be able to assimilate yourself. But on the other hand, if you actually adapt to the Indian culture and make use of it, turn it to your side. The adaptability is one of the key ingredients in your life's success. If you don't have the adaptability, you can never succeed. And so the adaptability is very important when you make a decision to move to another country or come back to your own country. That you cannot compare. You cannot compare India to US anytime. I've done that, and I don't do that anymore. But the thing is, it's very important. Sometimes you also have to voice your opinion to make sure that you give the right direction for the India also to grow to allow. Because I've seen best of the both worlds there and here. People are gems, no doubt about it. Indians are gems. There's no doubt about it, no question about it. And what we need is the education system needs to improve. Okay. I've seen in the US, I struggled for the first 1 year because I never had any practical bench experience.
Okay.
That gave me a hard time because once you go into a lab and try to do experiments, and you have to be thinker. You've got to be thinking of solving the problems, not creating problems. You've got to solve the problems. Solve the problems is a different ball game compared to creating problems. You can create hundreds of problems, but if you don't solve the problems, then the problems will be there forever. So what it gave me, the training gave me for 1 year, is that I need to be able to solve the problems, not to identify more and more problems.
Yeah.
And that kind of practical training is lacking yet in Indian education system. I mean, there are many reasons. There's nothing wrong about the Indian education system. I survived it. I'm glad I got my basic education in India. Right?
And you're glad because?
I mean, glad because actually gave me fundamental foundations of education, although not a practical applicability for it. I learned the practical applicability in other countries. So now I'm bringing that here to India to be able to teach the younger generations practical applicability of knowledge.
Okay.
Right?
Yep.
If you don't practically apply that knowledge, you can get nowhere. Yes, everybody has ideas. You have tons of, I'm a think tank. I can come up with 100 ideas. But the problem is I can't work on every idea that I have in my mind.
What is feasible, what is
You've
commercial?
Got to focus.
Yeah.
I can walk and say, hey Akash, this is the idea, that is the idea. But so what? Right?
Exactly, yeah.
But so I need to say, okay, this is the idea I need to focus on. That is how Reagene came into existence. Because finally I thought there is an idea that I can actually capitalize on, make it on. So the ideas keep coming, let them come in, but see the merits and demerits of each idea you have, and see is it going to be successful or not. For that you need to do a lot of background research.
Yeah.
And if you don't do that, you'll waste your time. So you do your background first to make sure that this idea will definitely catch in the next 5 years to take it to the next level.
And you have to only do sure things, right? You have to be absolutely sure.
Right. So, focusing is very important. This is what director of American Cyanamide told me. Very nice guy, and he said, "Hey, Manny, you have tons of ideas, but you don't focus on anything." Right? That struck me. Oh, focus. You gotta focus. If you don't focus, that you're putting your hands in 100 different things, but nothing comes out of it.
I don't think any known person has pulled off building his own company or inside another company wherein he has his, let's say, time distorted by 10 things or even 5 things. So what Buffett says on this subject is if you have 100 ideas, forget the 98 of them, probably do 1 or 2.
Yeah.
And that's the way you can even get anywhere in those ideas. The 98, people do not have enough focus, enough time to do all those. So you can only do one.
And also money, you need
money. Absolutely.
I mean, where's the money coming to? I mean, I just read somebody posted our biotech group 2 days ago. The 10 top startup companies in India today. You look at all of them, majority of them, I won't say all of them, majority of them are actually doing genomic-based platforms for the personalized healthcare. That's where the money is. MedGenome raised $200 million in that. Farcast raised 70 to $80 million on that. So Anu Acharya raised, I don't know, but maybe $2-3 million. I don't know. I mean, so if you look at that, the wave today is a personalized cancer genome. You need IT industry and the scientific industry to actually come together and build these databases to treat the patients with a personalized care rather than one size fits all treatment. So, I mean, back in olden days, this drug is available. Okay, he's got cancer, give this drug. But 98% of the patients, the drug doesn't work.
So getting back to let's properly, precisely define these stages of clinical trial. So the first stage is?
Phase 0, we call it, the concept going into regulatory preclinical development. You select the 2 or 3 good molecules that you have, and they have very good safety profile, compared to the efficacy profile. So you have a better efficacy profile than the safety issues. So you take those molecules into preclinical toxicology in animal trials. These are regulatory mandated trials. You even dose animals at 2,000 milligram per kilogram doses, and I've seen that, and I don't know the rationale for it, but these are required mandated studies based on. But today we actually apply the bioanalytical tools, actually understand the concentration in the blood and the tissues, maybe the response to the efficacy as well as toxicity, and see if it is a narrow therapeutic margin or a wide therapeutic margin. So safety is here, efficacy is here. So you have a lot of therapeutic margin. Even if I give a little overdose, still it's safe.
It's
right?
safe.
So that's very essential in the preclinical toxicology, that you establish wide safety margin. Where the trick is that you have to establish the dose to go to the first in human clinical trials. The reason is because in animals we take mouse, rat, dog, and monkey. We do species related pharmacokinetics, we call it. And this pharmacokinetics will tell you how a mouse is different from monkey, or monkey is different from dog, or something like that. Because you are using 4 different animal species here.
Correct.
If there are differences in that, you'll know that there will be major differences in humans too. So the pharmacokinetics is very linear and doing very well, because mouse is 20 grams, rat is 200 grams, monkey is 3 kilograms, dog is 10 kilograms. So human is about 70 kilograms on the average. So you scale it up and say, hey, my allometric scaling data is suggesting this dose to go into the humans for an effective dose. But the effective dose, we don't know if it is safe or very toxic, we don't know.
Does it linearly translate in terms of weight?
There are lots of ways to do it: body surface area, body weight, brain weight. People do different kinds of ways, but typically standard procedure is body weight. But I've done it, I think it would be 30% times I'm correct, 70% time I'm wrong.
So even a subject matter expert will go wrong, and somebody who's planning the study has to take that into account when he is budgeting for it.
Yes. This is where the informed clinical trial design comes in. Your design of the clinical trials depends on this data. Correct. It's not just checking a box that I did a dog toxicology, monkey toxicology, and rat tox. But how do you use that to design a human-informed clinical trial?
To sort of customize and learn
from it, and say, hey, this is what I need to do. Go into the first in human safe dose, right? So, so that's the first thing we do. And the toxicology studies done. In the toxicology studies also you're doing pharmacokinetics, we call it toxicokinetics. You now know which dose is toxic.
So this is phase 1 basically.
Phase 0 in animals.
It's still in animals, okay.
Yeah.
Phase 0.
So you get the data saying that, okay, this is the toxic dose, this is the blood concentration I have. The effective concentration is here or here. Basically, that you calculate it. And if the effective dose and the toxic dose doesn't have different margin, we say this is not a good clinical candidate, we don't go anywhere. But, so having said that, this concentration response is very essential in designing a clinical trial. And on top of it, the other thing we do is that humans definitely pharmacogenetically different. When you're doing animal trials, rats are kept in a cage. They have the best AC room, germ-free room.
Okay.
Humans don't have that.
So you're not isolating the candidate or the subject?
In humans, we all eat different food every day. You know, you eat pizza one day, another day you eat
something
chaat
else.
masala. So animals are given the same food.
Okay, there's more uniformity in...
So all these variables affect in humans.
Okay.
So when you're trying to take, to extrapolate the animal safety data, efficacy data, you need to take these into account when going into the clinical trial. So human metabolism is different.
Okay.
Okay, quite different. We don't metabolize like rats or dogs or humans, monkeys, sorry, in humans.
Okay.
So there's an enzyme called Cytochrome P450. It metabolizes about 30% of the marketed drugs today we have in the market. 30%.
Okay.
So there's another enzyme called Cytochrome P450, A4 in the liver, that metabolizes about 50% of the marketed drugs today. So together these two account for 80% of the marketed drugs metabolized. So if I'm developing a new drug, if it is metabolized by these two enzymes in the humans, and if you're taking the other drugs are also metabolized by CYP3A4, you're prone to have drug-drug interaction, serious side effects. This need to be also tested in the preclinical level.
So you're saying if you're coming up with a new drug, you have to look at the broad array of drugs which are already in the market and how many drugs for
how
which...
they interact with the new drug that I'm developing.
Correct.
Okay, so that's very essential. Number two is that these are cytochrome P540 enzymes. There are other enzymes, the transporter, drug transporters. A drug needs to be transported. Wherever it goes in the body, there are transporters, uptake transporters and efflux transporters.
Okay.
They take it and deliver it to the other side. If these transporters are different from person to person, the drug doesn't work. Some work only in few people, it doesn't work in other people. That's what we call it pharmacogenetics.
Okay.
About a dozen of us from various pharma companies. In 2008, we published a pharma white paper on pharmacogenomics. I was one of the co-authors on it.
Okay.
To show that CYP2D6 enzyme is very crucial in understanding, because CYP2D6 is an enzyme which 30% of the patients are metabolizing. So we do CYP2D6 polymorphisms. There are 100 different mutations in human populations in different ethnic groups.
When you take that drug?
Yeah, so there are, if you take some Caucasians of European descent, CYP2D6 may be about 5 to 7 copies.
Okay.
So they metabolize the drug 100% in fraction of a second.
Okay.
So your drug never reaches the target, active site, to be able to be effective. It's not toxic, but it doesn't work in those patients who have 7 copies of that gene, CYP2D6 gene. On the other hand, there are some people who have, about 10% of Caucasians are deficient of the enzyme, because there's one single cytochrome P540 2D6 star 4 mutation, they call it.
Okay.
And that mutation is there. There's no cytochrome P540 enzyme. The enzyme is dead. There's nothing is there. So you don't metabolize the drug. So now your concentrations are way up. This guy doesn't have any concentrations. This guy has this much concentration.
Yeah.
It's very toxic for these people.
Okay. Okay.
So pharmacogenetics becomes a very useful tool when you go to the clinical trials. And this is where the industry is trying to understand the genetic differences among the humans. Forget about animals. You and me are different.
Yeah, absolutely.
There's no animal model to define you and define me.
Yeah.
Right? And this is where the problem comes in selecting an animal model to define. So this is where a personalized medicine, precision medicine, is the talk of today.
Okay.
So we are, as Reagene Biosciences, building these personalized medicine precision tools to be able to work for you and work for me. So what I can do is go to the doctors and say, hey, this person has some cancer or some other disease. You want to treat him, but you don't know which drug you want to give him, because you're giving one drug, it doesn't work. You're giving second drug, it doesn't work. You give third drug, it doesn't work. So you don't even know which drug works for this patient.
Correct.
Right?
Yeah.
And you delay another 6 months like this, giving 10 different drugs, the patient is going to die anyway. So for us, if you give us a biopsy or even blood sample of this patient, we can understand what is the difference between in this particular patient. We can tell you which drugs to give the patient by using our in vitro human 3D bioprinted tool.
Okay.
So we'll put his patient biopsy cells into our tool and screen the patient. What is he deficient, or what is he having high levels? So we can actually do the genotyping, but also see which drugs actually work for him, and we can give a combination or a single drug, whatever that need. And doctor can now precisely give that medicine to make it work for that particular patient.
So in terms of phase 2 trials, we were discussing that phase 1 trials is first you look for toxicity
Safety,
among
yeah.
healthy patients. So what is the process of, what do we check in phase 2?
So phase 1, as I said, is a safety trial. Basically, you're looking whatever the animal safety trials we did in preclinical level, are they really true, or when our drug is safe or not first? Because even animal safety trials are done in healthy animals, not in diseased animals. And so basically, when you go to the first in human trial, you want to look at healthy subjects, not patients. So which means that if it is safe in healthy patients, and you're at least reproducing the animal work, and you're more comfortable that, yeah, this drug is not going to have any liabilities as we move forward into the other trials. Now the second question is that, yes, it is safe, but does it work?
Okay.
For the
When you
indication.
say does it work, you mean is it effective when you put it in humans?
Right, exactly. So the phase 1 trial also investigates what is the dose range that is available to you in terms of managing the therapeutic effect. Is it a
Okay.
wide therapeutic margin or a narrow therapeutic margin?
Okay.
So that gives you a little bit of an idea. It's safe over a wide range of doses. You can play around with in phase 2 trials better.
Okay.
Okay.
Yeah.
Otherwise, what happens is your phase 1 trial says it's narrow therapeutic, then your phase 2 window for the efficacy is very, very marginal. So you cannot play around too much.
Can you illustrate it with an example as to how you will
Okay.
define this margin?
Yeah.
Therapeutic margin,
For
let's say.
example, I was working on a project. It's a immunomodulator drug. It was safe, very excellent drug in the animals. No issues we found. We have very wide margin of therapy, safety, right? And then you go into the clinical trials. Clinical trials are segmented again. There's phase 1. In actually first you do single dose studies.
Okay.
Okay, escalating single doses, and making sure that in escalating single doses, you also take some blood samples to understand how much drug is actually in the blood circulating. That you
Okay.
can compare with the toxicology studies where the toxicology margins are there, and making sure that you have wide range of safety margin. And then next step is to do multiple dose studies. No drug is going to be effective in one dose. So you need to do multiple doses to see how long your drug is going to be safe. Are there any other issues? So you do at least 2 weeks, minimum 2 weeks, multiple dose studies. Depends on the designs and all depends on what is the therapeutic indication. You're looking for cancer drugs, maybe scheduled dosing, IV dosing, a lot of different things that happen in the clinical trial design at that time. So basically what you do is you are looking at multiple dose safety.
Multiple dose safety. Okay.
Right. And so this drug passed through the single dose safety studies very well. We did about 2-week trials.
Okay.
And in the 2-week trials, we found something weird. What happened was the drug, normal PK, pharmacokinetics, when you analyze the blood concentrations were normal in the single dose studies. Dose escalating, nice linear pharmacokinetics that you find. When we do the around day 12, the clinical trials were stopped because patients were having some skin rashes. These healthy subjects actually were having some skin rashes and other side effects.
Okay.
So immediately it was put on hold. Unless you understand what this means, we cannot take it forward. That's why
Correct.
it was stopped. The clinical trials were stopped. And I actually tried to investigate what went wrong there. So we looked at the blood concentrations of the drug.
Okay.
And the drug concentrations, as I said, they were linear in the dose 1, but when you look at the day 12,
Okay.
as we grow up the higher doses, the linearity actually gone, and highest dose is actually 10-fold higher concentrations of the drug compared to the day 1 concentrations.
Okay.
So that told us something, and we looked at a metabolite that was normally about 2 to 3% or something like 5% maybe, and we looked at the metabolite on a day 12 also. Metabolite disappeared. So we understood that there's something called metabolism is affected by the same drug. It's inhibiting its own metabolism, by which it's actually proportionally going up to a level, it's causing all the skin rashes and toxicities.
Okay.
That's the reason it was pulled out. And another drug, they have a backup. I looked at the structure, chemical structure,
Okay.
that has similar chemical structure, and I said, this is also not going to work. We
Work
did a lot of mechanistic studies on that, too, and the whole program was parked
Okay.
at the time. So these are the things that you see in phase 1 trials. And there's another example I'll give you, is that there was another compound. Same thing we were doing, the phase 1, the single dose data was quite good.
Okay.
But in this case, after day 10, the doses, the concentrations were 0.
Meaning the drug is nowhere to be
That
found.
looks like it's metabolized extensively.
Extensively, yeah.
And what we looked at was, we actually looked at the induction parameters, we call it. The same drug can induce its own metabolism. In one case, it was inhibiting its own metabolism. In the second case, it was inducing its own metabolism.
Okay.
Right?
Yeah.
So the induction, Cytochrome P450, there are mechanisms for that, the nuclear receptor-based mechanisms, by which actually it is inducing its own metabolism in the
Okay.
humans. So by day 12, it's not working. There was a very interesting case in one time. Some ladies were taking the prophylactic drug for preventing pregnancy,
Okay.
right? There's a drug that was on the market everybody was taking. There was another drug that came in, rifampin. It's a morning sickness drug or whatever. That they took it.
The night
along with
after
it.
drug, they call it that.
Huh?
That one? The night
Sure.
after drug, they call it, right? That one?
Yeah, probably. I mean, rifampin it's called. But what happened was with that drug. So that drug actually induces this enzyme so that the other drug, which is supposed to prevent pregnancy, is metabolized faster and it was not having any effect, all these ladies became pregnant.
Okay.
Right?
Yeah.
These are the things that happen in phase 1 trials, that we make sure that there are no hidden liabilities in that. And these phase 1 trials go very well, single dose or multiple dose studies with no incidences, and then go into the patients. The other thing is about you cannot directly go into the patients, it's unethical.
Okay.
Many patients are on other drugs.
Correct.
So you don't even know your drug actually works on the patients.
Okay.
Right?
So
So
how do you
directly going into the patients is very, very difficult, and it's not approved by ethics unless there is a real board, ethics review board
Ethics
actually
board,
reviews the entire clinical trial. But usually, except for some cancer trials, I've never seen many of the phase 1 trials go into the patients directly.
Okay.
Okay?
Yeah.
So that is an ethical concern, basically cannot directly go. And once you have the proof of concept established, in the sense phase 2 trial, you have very limited number of patients. And those patients, you may be able to pull off the drug, what they're taking, and give this drug to ensure that this drug works. If that doesn't work in phase 2 trials, all bets are off, the whole program is parked. And then, if it works in phase 2 trials, now you go to the phase 3 trials, which is where you have large number of
Samples.
patients that you're using. This is actually, you also use population PK/PD modeling to etc. to look at the effectiveness of the drug. And in fact, 90% of the phase 3 clinical trials fail, although a compound is effective in phase 2 trials. The reason is because you're going to population of 5,000 people, variabilities matter.
So the transition from phase 2 to phase 3, the success rate is 10%. Success rate is 10% is what you're saying.
Yeah, right. Phase 2, I know, phase 2, okay, if I have 10 drugs, that all pass through the phase 2, right? And I go into the phase 3 trials, I have only one chance of only one drug may actually pass through the
passing
phase 3
through
trial.
phase 3.
One, two, maybe, if you're lucky, 2 drugs maximum.
And the reason is you're testing it on a larger sample.
Larger sample population. This is where the statistics matter. Most of them are actually, the statistics show that it's marginal efficacy or no margin of efficacy. So
at baseline efficacy, let's say.
right. So baseline efficacy, and many times that doesn't work in terms of pulling it to the market, and that's how all those phase 3 trials fail.
Usually,
Very
what is
rarely, if safety is an issue in phase 3 trials, because we've already looked at the safety in phase 1 and phase 2 trials.
but what if we do encounter some toxicity?
We do encounter in some cases. I mean, I would say in phase 3, 80% is efficacy failures and 20% are safety failures.
Okay.
Approximately.
There are still failures though.
There are
Safety failures are still there, but it's just less in number.
Right. Then once you got it and you go to the FDA, and FDA sometimes may have more questions, and then you may have to do more clinical trials. It depends on what FDA feels. And if they bless you and say, hey, you can put it in the market, now you have a phase 4 segment. It's already being given to the general public patients. Now you're actually talking about millions of people are taking the drug.
So that's the phase we are in right now, at least in COVID. We are in phase 4?
No, we are still in phase 3 because we don't have any drug approved yet.
So the thing that is making the rounds, AstraZeneca, or let's say some of the Pfizer is making some news that they are ready with the Moderna. They're making some news saying that they are ready with.
I mean, there is.
So what is it that they're trying to say?
Well, I mean, from what I'm reading, okay.
Yeah.
I'll just. What they're reporting is that the 95% effectiveness appears to be that it actually reduces the symptoms of corona infection. Symptoms.
So this is reduction of symptoms. And the fact that it's an infectious disease, that is not being reduced.
Yeah, right, exactly. So the thing is, we still do not know these vaccines prevent the infection.
Okay.
Okay?
Yeah.
So that's the thing that they haven't answered, because you know what? I don't blame any of these guys, because you're fast tracking all these vaccine trials. To do everything, normally a vaccine trial will take 5 to 7 years. And
We
now
don't
when
have
you want
5
to
to
get a vaccine within 1 year, you have to do studies that will give you some comfortable data to be able to do it. As long as they're safe to the patient and
Yeah.
they reduce the symptoms, hopefully you can do more trials as vaccine is being given to subjects.
So how far do you think, in terms of your timeline, that we are close to getting a vaccine in our hands?
Well, we may do get an approval in the first quarter of next year, hopefully. That doesn't mean we are out of the woods. I'm saying it's because we still don't have a lot of questions answered in terms of their vaccines are supposed to prevent infection. They are supposed to be, for example, when you get a vaccination as a child, it's actually prevention. Next time you don't get infection, because the antibodies are already there, or antibodies are boosted when the infectious agent attacks you, and that neutralizes the
Infectious
agent. But here what you're talking about, most people in the COVID, at least coronavirus 2 cases, you see most of the people, vulnerable people, are 65 and older.
Okay.
They have comorbidities. A lot of people have diabetes, atherosclerosis, and arthritis, and many other diseases.
Okay.
So with these comorbidities, they're also taking other drugs,
Okay.
right?
Yeah, got it.
So what happens is that most of these cancer, any disease you take, they are immune diseases. Now you're actually attacking immune system to stop the cancer progression or spreading. So basically the thing is, immune system is compromised in many of these
Patients.
Patients with comorbidities. And this is where there are different schools of thought here. Some people are saying that we should go straight into the older populations with the vaccine, so that they are the vulnerable populations. But the question comes in, they are also immune compromised patients. Is the vaccine going to boost the immune system to, especially the adaptive immune system, to produce the antibodies?
So I was of the view that you are able to stabilize the immune system and prevent it from deteriorating further. Are there ways by which you're boosting?
That is called, you're using immunomodulators like dexamethasone, and also something like hydroxychloroquine, which is controversial, and people are using other drugs too to bring it down. But what these drugs do is they actually stop the immune system from producing the inflammation.
Correct.
Okay?
Yeah.
So the inflammatory system is subdued. So you don't get this. Inflammation is another toxicity phenomenon.
Okay.
Most of the inflammation is not good for health.
Most of the information.
Inflammation.
Inflammation, sorry.
Yeah, inflammation is not good for your health.
Correct. But inflammation, and that is generally known, right? Even athletes, when they recover, they want to reduce inflammation on a regular basis.
So that inflammation actually is problematic as we grow older and older. And so in this COVID cases, looks like inflammation is very strong. The cytokine storm that follows the infection is the reason why a lot of people are going into the hospitalization.
Intubation.
Hospitalization into the.
So essentially, inflammation is broadly wear and tear on the body?
Actually, broadly, it has a lot of wear and tear. Okay. There are lots of inflammation is also linked to cancers.
Linked to cancer.
Yeah, predisposing people to cancers.
Okay. So now we understood that this drug might not be a panacea, but given the situation we are in, this will at least bring down.
It might at least stop deaths or decrease the number of deaths, because if you are reducing the symptoms, they don't go into the latest. You can also use these immunomodulators like a dexamethasone to also stop the cytokine storms. So you can make a patient feel better and not go into the fatal state.
And apart from smallpox, we haven't gotten any vaccine, which is smallpox is the only disease that we have eradicated from vaccine, right?
Right, yeah.
All the other diseases we have sort of eradicated, but not really. There are still incidences here and there. Am I right on that?
Yeah, smallpox, I think when I was into late 20s and I got once. And it was actually, it itself immunizes your body, you don't get it again.
Okay.
Okay? That's the way it was. I didn't take any drugs really to tell you the truth. There are no antiviral drugs for that. So basically that's self-curable. It's not life-threatening unless in some cases people are malnutrition and all the other things. So in many cases people, the people who got infection, they'll come out. I think chickenpox is more dangerous than smallpox.
Okay.
If I remember correctly, I have to go back and look at it.
Yeah. And you think like natural immunity is better than, let's say, drug-induced immunity?
Of course, naturally it is better, natural immunity. So if we all can survive, like smallpox, if we all can survive the virus, then the natural immunity will prevent you to get the disease again.
Okay.
Right?
Yeah. So any immunity is not comparable to the natural immunity that human beings already have.
Right. Yeah.
And so we covered the fact that what is in the news should be taken with a grain of salt.
Grain of salt, but it's a good news. To me, it's a good news. But more need to be done so that we can actually have reduce the fatal incidences.
When can we completely put COVID out of the picture? How far?
Without any drug or vaccine or anything. If I compare it with the Spanish flu in 1918 to 20 that killed
50 million people?
Yeah, 50 million people or something. I think this is a milder one compared to that because we don't have that many deaths compared to it. But again, the people are talking about second wave coming in. We don't know how damaging it is, how strong it is, and so we still don't have answers for it. But if you have to wait until the herd immunity, natural herd immunity,
okay,
that's going to have a lot of casualties. Maybe 5%.
All the people with comorbidities
Yeah,
will probably...
comorbidities, 5% definitely casualties will be there. If you look at 5% in 100, in 1 million, you can calculate it.
Yeah,
so
that's a huge number. And
you
if you have 7 billion
have
people...
7 billion people in the world, you can calculate.
That's a lot of people.
Lot of people. That's a lot of people. So we don't want to go calculate those numbers.
Yeah.
Right? So vaccine is one way to get the herd immunity.
Okay.
Without casualties.
Okay.
Okay? Got it?
Yeah. But I was thinking it's called herd immunity if herd immunity need not be naturally induced, correct?
Herd immunity is need not be natural, but it depends on how mild is the virus. Okay. If the virus is too life-threatening, natural immunity doesn't help you. So you do need a vaccine to get yourself immune to the virus. That's not going to kill you. Vaccine is not going to kill you unless if it itself is not safe. But usually vaccines are supposed to be safe. So vaccines are supposed to stop you, right, from getting killed by the virus. So what happens is that now you're immune and you cannot infect anybody else.
Okay.
Got it?
Yeah.
So that's where you have herd immunity already in place. So in my opinion, if the vaccine is given to the people between 18 and 40, of course their immune system is very good. They're also afraid to go out. We don't want to tell them that, okay, go, you're a healthy person, you can go and get immunized by the virus itself. That may be dangerous. So instead of that, if you can give 18 to 40, these are the healthy people with a good immune system still, give it to them. And if they're all immunized, me as an old age guy with vulnerable comorbidities will be much safer. I will win without vaccine.
Okay.
Got it?
Okay.
So
Got it.
that's the way I'm looking at it.
Understood. But there'll be still danger for people who have these comorbidities.
No, because it has to spread from other person.
Yeah, yeah. And
Right? It's a human to human infection.
And
So the other human is not able to infect me. I'm safe.
yeah,
Even if I don't have a vaccine.
got it. So that essentially is sort of herd immunity.
So that's sort of herd immunity, exactly.
And is there any truth to this idea that kind of is rumored that some of the vaccines cause autism?
Well, I don't want to go into that. It's because
Is it bordering on the lines of
well, I
conspiracy
mean,
theory?
see, there are claims, but the original article that came out on that has been disputed heavily. That it was kind of people said that's not true. So there's no proof this way or that way that vaccines cause autism. We know every kid is getting vaccination today. You know, I mean, I had to take my daughter several times up until age 10, and she got so many vaccines into her.
Yeah.
So I mean, unless the kid did, himself or herself has some other genetic defect, I don't think autism plays a major role in vaccines. So
Okay.
they themselves have some defect, birth defect or something, genetic defect. We don't know. So it's really, I can't say that vaccines cause autism or not. It's a controversial subject altogether.
Okay. But is it like 50-50 or is it like 90-10? Like only fringe people are talking about it?
Don't know. I don't know.
We don't know. Okay.
I don't know. I know a few cases. Kids have autism, and their parents claim that because of the vaccination they got autism. But I can argue with them because I don't have data to tell them that vaccination doesn't cause autism.
Most of these are anecdotal evidences.
Huh?
Anecdotal evidences. They're here and there, but they're not there in droves.
Yeah,
and
so
numbers.
see, the thing is, you hear something.
Yeah.
The problem is the public is now at disposal of a lot of Google stuff.
Information.
And
yeah.
they Google it and they think, I mean,
They
I
might
don't
not
want,
get the context because they're
Right.
not part of the system, or let's say they don't know the entire body of literature that surrounds that
So
subject.
this is the difference. You know, you can read a lot of Google, but somebody, I'm not criticizing Google knowledge is not good, but what I'm saying is that somebody like me can go into the PubMed and read the publications and actually go into the real data and digest it myself and make an informed decision for myself whether that claim is true or not true. But in Google, it just generalizes everything and tries to put it there, which a layperson cannot really analyze that data to a level that I can analyze.
Okay. So coming back to, let's say, your product of 3D printing these systems, or let's say simulation which simulates human interaction with the drug. When a human intakes the drug, the simulation that happens in your body is being sort of simulated in these 3D printed parts.
Yeah.
So that's what you guys are producing,
So-
and how are they different from, let's say,
Right.
animal models?
So we all have physiology. If you take a drug into the orally or IV, you don't know where it's going, number one.
Okay.
Yeah, it's going somewhere in the body. The advantage is that what you need to know is that is it safe or not, number one. And number two, you need to know whether it's working or not for the disease or indication, whatever you're talking about. These are the two things. I mean, as long as it is safe and it works for the indication, who cares? Everybody is fine with taking the drug, right?
Yeah.
Even if it doesn't work, you're safe, right?
Yeah.
So that's the concept. You have drugs like paracetamol, metformin. These are all wonderful drugs. They've been there for a long time.
Okay.
And they're fairly safe. I mean, even paracetamol at very, very high doses, unless you take 3 to 4 grams per day, it's not toxic to your liver, right? So
Yeah.
who's going to take 3 to 4 grams per day? No. I mean, the standard dose is even 500 milligrams. Even FDA recently said you should not take more than 350 milligrams if it is combined with some other drugs.
Okay.
So these are all safe drugs. Basically, we know quite a few drugs. But where the problem comes is this is where I want to go to the post-marketing effects.
Okay.
There are several drugs that were marketed.
Okay.
I would say if you go to Pharmacopoeia, maybe you can fish out about 3,000 approximately so drugs that are already on the market.
Fine.
There are many drugs, about maybe 3 to 5% drugs, have post-marketing black box warnings or withdrawn from the market.
Okay. Withdrawn from the market, is
Withdrawn
it?
from the market. I'll give you one example. I was in 1995. We were working on some programs, and at the same time, suddenly Roche released a calcium channel blocker called Posicor. Mebofredil is the trade name, I think. They released it into the market as a wonderful, what is that, the blood pressure monitor or something. And that drug, within 1 year, I can't remember the number, but more than 3,000 to maybe around 5,000 patients died
Because of the drug.
because of the drug. And the clinical trials didn't pick it up. Phase 3 clinical trials didn't pick it up, and phase 2 clinical trials didn't pick it up. Phase 1 clinical trials
There was
didn't
not
pick it up.
even an inkling or some breadcrumb that you can sort of go back
So
and check.
even animal studies didn't pick up anything, right? So when they went back and looked at it, the same patients, mostly geriatric patients, they were on other 4 or 5 other drugs.
Okay.
So there's a huge, serious drug-drug interaction problem for this drug, and with which a lot of people are going into cardiac arrhythmias,
So
sudden
you-
heart attacks.
You're saying this is one way by which you can simulate the interaction, or you're
Yeah, that's
trying
what
to-
we do at preclinical level now to simulate the interaction. If they have any liabilities of the interactions, we don't take that into the clinical trials.
How many types of people do you have in terms of, let's say you want to look at populations and look at incidence of the diseases that they might already have,
Right.
and they would be taking some drugs, and based on the, let's say, decreasing ranked order of these diseases, you want to check interaction with all of them, right? Your system should do
Right,
that.
yeah, we don't do that way. I mean, it's very difficult to do that.
Okay.
At a preclinical level and a clinical level, we have a streamlined programs. So I said cytochrome P450, 2D6, and 3A4 are the major enzymes that metabolize drugs.
Okay.
So I want to see my drug is a potent inhibitor of these enzymes.
Inhibitor.
Inhibitor.
Okay.
Because cytochrome P450, 3A4 metabolizes 50% of the known drugs on the market. Cytochrome P450, 2D6 metabolizes about 30% of the known drugs on the market. CYP2C8 and CYP2C9 are also involved to some extent in other drugs. But I focus on these 2 enzymes first. The reason is that if my drug is a very potent inhibitor of these 2 enzymes, then the co-medications,
Okay.
metabolism may be inhibited my drug, the new drug that I'm trying to introduce. And those co-medications, if they are inhibited, their metabolism, they become toxic.
Okay.
And this is why we do testing on these 2 enzymes first, and to make sure my drug, null drug that we are putting into the humans, is not a liability in terms of the inhibition of these 2 enzymes and interacting with other drugs. There's the Posicor or some other drugs, we have known that some people have actually are taking them at breakfast. They were taking this drug and they were having breakfast. Along with it, they were also drinking grapefruit juice. Grapefruit juice is supposedly a healthy drink.
Okay. Was there an
better,
interaction?
better than orange juice,
Okay.
right? So a lot of people actually were drinking grapefruit juice at breakfast. And they take the drug too in the morning, right?
Yeah.
A lot of people died.
How many?
I don't even know.
But quite a few?
Quite a few. And this is when they did the analysis, what happened, these patients, why they died. they are understood that the grapefruit juice, there's a chemical in the grapefruit juice called 5-hydroxybergamotin.
5-hydroxybergamot.
Bergamotin.
Okay.
That is this very potent inhibitor of cytochrome P540 enzyme.
Okay.
So that's inhibiting, the drug they are taking is going to a level high concentration that killing the heart.
And won't people who, when you do phase 3, won't there be people who will be consuming diversity of dietary habits?
That's a good point. Actually, typically
And that 5,000 number should be able to cover the gamut of dietary habits.
because these happen in 1 in 3,000 to 5,000, these interactions.
Okay.
So when in phase 4 trials, when patients are taking, millions of people are taking these drugs. That's
Okay.
when you notice these interactions. You don't notice in phase 3 also. The reason is because patient number is still statistically lower compared to post-marketing trials. So you cannot do a clinical trial for...
What if there is a failure, and let's suppose some drug passes phase 3 and there is a failure in phase 4 due to, let's say, dietary interaction. Now what happens to that drug? You shelve it completely, or you take it back to zero, or you start from phase 3 onwards?
So I cannot regulate grapefruit juice.
Correct.
FDA cannot regulate grapefruit juice. You cannot remove the grapefruit juice off of the market. The only thing they can ask you to remove is your drug out of the market.
I mean, at phase 4, it's still being tested, right? They are not like selling it.
No, it is selling it. So it's
phase 4 is
phase 4
already
means
sold.
they are already on the market.
Okay.
It's a pharmacovigilance you're doing at that stage.
Mm-hmm.
Okay.
Yeah, so it's like an arm's length distance, but you
Right.
still worry about the efficacy
Right.
and the results and stuff.
So
What if something fails in phase 3? You go back to phase 0 again, or you start from phase 3?
no, you cannot use that drug anymore because it's already failed drug. Okay.
And is there something called a failure event, or is it the intensity of failure?
There's another way to look at it. You know very well Viagra, right?
Yeah.
They were using for another indication, I think blood pressure relaxation thing, because it's a nitric oxide stimulator. Nitric oxide does the vasodilation. They were trying to decrease the high blood pressure with that drug. That was the reason they were doing clinical trials.
Okay.
Pfizer. That clinical trial miserably failed.
Okay.
Okay, the patients were not having any vasodilation in the blood. Sort of thing. But how they discovered was that this drug was actually patients who were taking it and they refused to give the leftover medication.
The patients who were taking it were refused to give the leftover medication.
That's what I hear the news. I mean,
And
this
the
is all
reason they refused?
is that they have a better sex life. That's it.
Okay.
Okay.
Got it.
So
Yeah.
got it, and so that's the reason. Oh, okay.
Yeah.
We have a drug for enhancing the.
Erectile dysfunction, essentially, but.
So that's how you discover sometimes it's accidental. So it's called drug repurposing because this drug didn't work for one indication, but it's working for another disease. So you may cut short because of this drug is not working for this particular disease. It may work for another disease.
Okay. So in terms of timeline, are we looking at another year of COVID or another 2-3 years? I mean, this is a repetition of the question.
Well, I mean, I'm hoping we'll have.
Best case scenario, worst case scenario.
I mean, I don't want to predict it because all the predictions are, even Anthony Fauci has been criticized that he's wrong many times. So these predictions, without enough data, we cannot predict any outcomes. But in my opinion, I think 2021 we'll still have to be a little bit careful until, I mean, I don't know about India. India, they're saying the cases are all coming down, we are fine. But at the same time, I'm hearing Italy, US, UK, Germany, everywhere, the cases are again going up, second wave is coming and going up. So it's very hard to say What is the reason? And some people are saying there are mutations, new mutations are coming
There
out.
are no cases in China, right?
I don't agree with that.
But
I
there are
mean, we don't know what's happening inside China.
Mm-hmm.
Okay.
But I mean,
I
these.
just read an article. I don't know how far it is true. There's Chinese are saying that this coronavirus actually came from India in June 2019.
India.
India
How?
originated in India. They're giving some reason saying that it was a milder form of coronavirus in India. Somehow it entered China and mutated and became deadly.
But there is no evidence for that, right?
They're saying that they say that they've done some phylogenetic gene mutations and everything. They were looking at the phylogenetic tree of the coronavirus, and they looked at the coronavirus that is right now in India, and that they say that the mutations for coronavirus too may be more deadly than the virus we have here.
Okay.
So they're trying to justify it somehow that it came from India, not from China. So they're shifting the blame to India.
Alleviating all responsibility.
Right, so it's politically motivated,
convenient.
motivated argument. So,
Lot, yeah. So now, but the broad idea is like, how does a country like India procure all these vaccines? Is it like you procure it from private players? This sort of an event has never happened before, right? The adult immunization.
This is one other the thing about logistics of distribution of the vaccine. Pfizer vaccine requires cold chain at -80s to be distributed all over.
-18 or
Minus 80.
80. That
Temperature.
seems like a cost.
Whereas Moderna vaccine requires 4 to 8, so refrigerator is enough to
Moderna.
distribute it.
Hmm.
So this becomes logistically very difficult to distribute these viruses at different parts of the world, number one. Number two, the Pfizer vaccine, President of the United States will say first come, first serve is Americans.
Obviously.
Right? So make all the doses only for Americans now. We'll think about the second. I don't know what is the manufacturing capacity right now for these vaccines. Is it 500 million doses or 1 billion doses? We don't know. I mean, I think maybe a million doses or maybe 2 million doses at a time. We are lucky if maybe 10 million doses at a time. So the manufacturing capacity defines how far we can reach out to the entire populations in the
And
world.
who pays for this?
Huh?
Who pays for this?
Distribution. So there are programs, of course, R&D by Pfizer or Moderna, Oxford, were funded by their own money.
Yeah.
Okay.
Yeah.
But there is a government has agreed to pay some billions of dollars to, if in case this Pfizer works, this vaccine works, Pfizer vaccine or whatever it works, then for the manufacturing and distribution to accelerate that process, we'll give you some $100 to $150 billion or something. The government, it's called Project Warpspeed, introduced by Trump administration.
Mm-hmm.
That only if they have a vaccine that is ready to be distributed, manufactured and distributed.
And they'll bear the logistic cost, essentially.
That is how some of the government funding is also coming into it. I believe the same thing is happening for Bharat Biotech vaccine to Modi's government. Maybe.
So, is there a vaccine in India which is comparable to, let's say, Moderna?
Don't know. I mean, all these vaccines are different vaccines. And they're not the same vaccines.
Okay.
Moderna vaccine is an mRNA vaccine,
Okay.
whereas the Pfizer Biotech is a recombinant spike protein vaccine. Same thing with Bharat Biotech also have some spike protein, I think, alternative, but there's a different way of cloning it compared to Pfizer's. So they all have different vaccines. It's not the same vaccine exactly with the same properties.
And do they go ahead and try all of them simultaneously, or do they decide
No,
which one works?
what will happen is this. This is only a single protein antigen,
Okay.
right? Spike protein is only single protein. That is the major protein that binds to the ACE2 receptors in the lung, and
Okay.
through which actually it gets access into the lung cells.
Okay.
So if it doesn't bind to the ACE2 receptors, virus cannot get into the cells.
okay.
We can assume that. So if I have very low ACE2 receptors in my lungs,
Hmm.
I probably won't get infected. I don't even know that, right?
Yeah, yeah.
Because
So that
ACE
is one of the technique that is being employed.
people are looking at ACE2 receptor polymorphisms right now on some people, but it's still a lot
Early
of
days.
research to no idea people for people. But what they're saying is that ACE2 inhibitors, for example, there are drugs that inhibits also ACE2 receptors. If you inhibit the ACE2 receptors, maybe virus cannot penetrate. But still, that's in research stage. We don't know where they are at. But there are also counter arguments and theories around it. ACE2 receptor is the only one, but there are other receptors or proteins on the cell surface may also access, have access to the spike protein to be able to allow the virus to enter the cells. So we still
Okay.
don't know a lot of that information because still it's in a molecular biology, many times you think this is what's happening and 6 months later somebody publishes something else. No, this is also happening. So it's very difficult to say ACE2 receptor hypothesis is the only ultimate, the theory. So people are looking at ACE2 receptor right now. I mean, there are publications that already look at how many people have ACE2 receptor polymorphisms, thereby they have very lower levels of ACE2 receptors. So their infection rate is going to be lower compared to people who have high receptor abundance in their lung cells. So basically you're looking at these receptors abundance also in patients or people in general populations. If I come to know that I don't have ACE2 receptor, a very little ACE2 receptor, I can just go walk around, right?
Okay. Yeah.
Right? I don't need a mask because I don't get infected.
Okay.
But that's taking, a lot of risk.
Yeah.
Right?
We know that. I don't know that
So-
for sure. Now, given this scenario, we cannot be sure of the dates. We cannot be sure of at least the years. Like if the Moderna thing works out properly, it'll still take a year before the rest of the world catches up.
Yeah,
Right?
everybody to get it, you're talking about 2 years.
2 years, easily.
Easily. Everybody to every corner of the world
Will
getting
they
it.
sell this, or what is it like? They will give it for free?
It depends on the government programs. If the government subsidizes it, they may become free or even very low cost. Insurance maybe also can chip in and pay for it. So it all depends on the government, insurance companies, how they come up with a plan to distribute.
How many dosage are they coming up with? I mean, it's multi-dosage thing, right? It's not like one
Right.
dose.
I don't know. I mean, each one is talking different numbers. I really don't know, to tell you the truth. How many doses are there for each vaccine to come out in how many batches or anything. Each batch also need to be verified. AstraZeneca vaccine, they were talking about, they had a manufacturing defect. In the
Okay.
sense, they made 2 doses, right? They said this is 1 dose and this is another dose. You have to give 2 doses for the vaccine to be effective. In some patients, the first dose they got is not full dose, they got half dose.
Okay.
That's because of manufacturing defect. Manufacturing people didn't calculate it well or something fell apart. So it's only half dose they got it. Okay, so
This is all happening in the public eye, right? So wouldn't they have the amount of scrutiny to go through it properly?
unfortunately, because you have so much rush and race
Hmm.
to the
They forgot to do it.
somebody overlooked it, is basically. But what they are claiming now is that, in fact, that lower dose is far better than the actual full dose 2 doses.
Now,
In
to
terms of-
justify.
Justified,
Yeah.
basically. So these are the things that you need to look for. Make sure that the manufacturing
Capacity.
capacity and also the quality
Yeah.
Manufacturing quality is very, very critical,
Hmm.
highly critical. Many companies are shut down by FDA because of the poor manufacturing
Facilities?
practices,
Practices
and
and facilities. Yeah.
operations. So FDA is not going to let you distribute that batch if there's a manufacturing defect in it.
So, should a drug manufacturer in India, I'm sure there is a FDA parallel in India, ICMH or.
ICMR, DCGI are the
CMR.
ones that are trying to do. Uh,
But it's not as powerful as FDA.
Yeah, I wouldn't say that. Say the FDA. If you walk into the FDA, there are lots of 3,000 PhDs, doctors reviewing the clinical packages.
Okay.
Okay, and we don't have that kind of a scientific rigor in the reviewing the packages in India with DCGI and ICMR.
How
So-
many people are we talking in India?
I have no idea. ICMR is the major body, I believe, is reviewing all the clinical packages that submissions. I
So
still
it's
don't
less
know.
than 1,000 people, maybe.
Yeah,
Okay.
but the thing is, these are dedicated people who don't have any other job except reviewing the packages that coming into the FDA. And I know many of my friends who are actually done 10 to 20 years of pharmaceutical research. They were taken by FDA because these guys have experience in pharma industry.
Correct.
And ICMR doesn't have any experience in pharma industry.
Because pharma is a new industry here.
Right, so pharma industry experience is required because we have gone through the concept to the marketing level. Each step there are people experts.
Correct.
There are preclinical experts, drug discovery experts, clinical experts, quality assurance experts, manufacturing experts. All these people come together, regulatory experts come together and do this job, handing over, like baton, handing over. Drug discovery scientist hands it over to preclinical toxicologists, clinical toxicologists hands over to clinicians, and clinicians with the clinical pharmacologists and clinical safety experts, clinicians. They all come together at different stages, and that is how the drug moves from all the way from drug discovery into the phase 3 trials and to the market. So
Okay.
that kind of a rigor, I don't see it in Indian clinical research or clinical trial management, or even in DCGI, ICMR. The one reason maybe India is not able to get into the innovative drug discovery is one.
And the reason is?
The reason is because we don't have the end-to-end experience. From all the way from the concept to the manufacturing drug discovery programs. These are the things that you learn when you're actually being on the project managements in big pharma companies like J&J, Wyatt. That's a blow. Even drug discovery was done by Reddy's, Lupin, but I think success rate is nothing really because they are very good companies. They had very good people who are in the scientific teams and heads. But the knowledge of transitioning from one stage to another stage is data driven. And you need real good experts to be able to see where are the liabilities, where are the positive indicators. So you do need a kind of system that review everything. I alone cannot review the entire package and say, hey, yeah, it's okay.
Understood.
Right?
Yeah. And how long do you think, what kind of work do Indian pharmaceutical companies do? Most of them are in generics business.
Right. The another thing is that the financial muscle.
Hmm. Yeah.
A drug, as I said, and in the US, takes on average $2.5 billion to make a
Okay.
drug into the market. I think if you look at the market cap for any company in India, not even $5 billion market cap.
That's
Right?
Yeah.
So with that lower market cap, who's going to be able to fund a $2.5 billion
Project.
from concept to finish?
But I'm assuming that's also cost of living, things like that. It's 1/5, probably 1/10 of that, right? But even 1/10 or 1/5 is still a big number.
It's expensive. It's still expensive in India. The reason is because you're importing everything into India. Even the raw materials, you're importing everything into India. So all the instruments that I use in my lab, they have to be imported from US and Europe, Thermo Fisher or anything, or Japan or something. They're not giving me at 1/10 of the price, those instruments.
And why hasn't the pharma companies, associations lobbied for removal of the exchange duty that they apply once it crosses the border?
There are some of those kinds, but they're still expensive to me. See, if I have to buy a 3 to 5 crore mass spectrometer, look at the funding I get. You know, Biagra fund is only 50 lakhs. 3 to 5 crores mass spec, I can never, as a startup company,
Yeah.
it's impossible for me to do it. So, more and more business if I do out of India, then if I get more and more business from there, then slowly I can ramp up my revenues and capabilities to be in par with American.
The idea is like if you have to suggest and give some tips to
kids,
young
right?
biologists,
So,
biochemists.
I have mixed opinions on that. I myself ran out of the country
Yeah.
for my own opportunities.
Okay.
Right? I cannot ask them to stay in the country and India is great and we can do everything. But the thing is, when you go out of the country, your ability to see things differently improves a lot. That's a very important thing. You know, not just don't stay in one place, in my opinion. There are lots of people who want to stay at one place all their life. Family reasons, mostly family reasons. Family that is dependent on them and other commitments that they need to be there. But apart from that, you know, India is growing now. The opportunities are more than what I used to have when I actually graduated with my master's in biochemistry. So there are more job opportunities to be able to take. For example, a startup company, it depends on individual level of competence. I've seen some people who are extremely competent with just basic PhD in India with no industry experience. There are other people I've seen that they really need 5 years of training on the bench and other things to be able to think through the programs. So it all depends on individual level. And I'm not saying that everybody has their own interest, what they want to do it. So startup companies, for myself, I couldn't venture out to start my own company for a long time. The reason is because I wasn't comfortable that the idea that I chose is actually an idea that will
Work inside a big company?
work out. I mean, make me,
A huge amount of money.
not just money, even bring the revenues.
Bring the revenues, yeah.
Right. I mean, you need to survive. You know, you have to pay your employees, you have to pay rent, and all these things are part of the. So if you don't have that skills, not only just doing science, you need management skills.
Ability to manage money, hire people.
And
teams.
manage people. You know, and sometimes if you're working with 50 people to 100 people, you know, you also need to manage the fights, little small cat fights among them.
Absolutely.
So all these things comes with a management experience. You just don't grow into it overnight. And there are some people who can grow into overnight, but it depends again on how good you are at and management skills, business skills, not only scientific skills. So people have to acquire them over the time. As I said, Dr. Fred Tonelli said, first go and learn what are the basics in India, rather than just starting a company, going back to India and starting a company. Very wise advice, right?
Yeah, absolutely.
So
Otherwise-
we need to really take a step back. Am I ready to it?
Yeah, that is true.
Right? So, and also you should be able to accept failures. Nothing you touch, everything is going to be a gold mine.
Yeah, absolutely.
You need to make sure that there are coal mines too, not gold mines. So,
Yeah.
right?
And how do you deal with, let's say, setbacks? Is there I don't think there is any recipe to get out of setbacks, but people do encounter, let's say, on an entrepreneurial journey.
Positive attitude. Every time you have any setback, okay, I'll move on to something else. Right? It's part of life. You know, I mean, you'd never give up.
Yeah.
And that is the persistence is very important in life. It's not nobody becomes successful in one day. Even Dr. Reddy says that he had to do 3 ventures before he was successful with Dr. Reddy's. So a lot of people are saying that. I mean, we are not overnight success with one venture. We had 2 to 3 ventures to understand how to manage a company or business that we are starting at. So you never give up and keep going on with the failures. It's okay, failures are fine in life. There's nothing, even if you fail, but you've attempted to it, right?
Correct.
You didn't give up, you attempted, you have an idea, you wanted to see if that idea can make something to you, mean something to you. But do that attempt, you know, I mean, don't say, hey, a lot of people will also discourage you in the process. But
Correct.
you have to believe in yourself.
Correct.
It's good you get advices from other people, but at the same time you also need to know your own strengths and weaknesses and where your strengths are. If you don't know your strengths and if you don't know your weaknesses, if your weaknesses take over your strengths, then nothing.
So, in terms of general people who are interested in this field of clinical trials, because they, like people who are under 18, look at the entire thing that is happening,
Right.
and they'll be very interested about clinical trials. There'll be a lot of fascination about vaccines, drugs.
Yeah, right. Clinical trials.
Are there any tips for them as to how they can follow up on all this information and have a career?
Clinical trials, you need to understand how, because these are ethical trials, so you cannot just go and run a clinical trials on anybody. You need to make sure, if I'm recruiting patients, you have to take consent from them first. There's a consent form, and without consent form, you cannot do a clinical trial.
Correct.
That's the most important thing in ethics of starting a clinical trial. And also, there are lots of things that exclusion, inclusion criteria. Sometimes you have to make very informed decision on how to exclude patients that you don't want in this trial. They may be good candidates for your drug, but maybe the other things, somebody who has a kidney problem, you cannot recruit them into the trial. Because
and if you have not incorporated that in your inclusion criteria,
excluding criteria, then that patient is going to die tomorrow. So
on top of it, you'll botch your own trial.
right.
Yeah.
And so those are a lot of things you have goes into that, and you have to understand all the nitty-gritty details in patient recruitment, the exclusion, inclusion criteria, and also who are the right population to do a clinical trial. If you don't recruit the right patients, for example, I'm talking about breast cancer, right? In breast cancer, there are about 20 different gene mutations.
Okay.
Right? So if I understand the patient, what mutation he has, my trial will become more successful because I can recruit only those people with that particular mutation rather than recruiting all the breast cancer patients. My clinical trial outcome will be nothing.
Understood.
Okay? So this becomes where precision and personalized medicine is where the genomic data is now incorporated in patient recruiting as well.
Okay.
So
How much of your, let's say, your success, if you can attribute that, is to, let's say, hard work, creativity, luck, if you have to give percentages?
I don't think I'm a very intelligent and creative person, to tell you the truth. But I work very hard. But I also try to understand the fundamentals. If you
Okay.
forget the fundamentals, you'll never do it. Even Elon Musk, you probably sent that video one time to me.
Yeah.
Elon Musk also said, fundamentals are the basics. If you don't know the fundamentals, you can never build business.
Correct.
So keep abreast of the fundamentals. Right?
Be it business or the, let's say, the domain.
So I'm a very fundamental person. I go to the fundamentals
And
to make sure what I'm doing is right. Fundamentally, it's right thing to work with. Scientifically and fundamentally, the right thing to work with. So that's where I put my hands only in things that I understand in depth. If I don't understand in depth, I don't put my hands into it.
Okay. And also, are they life lessons that, having gone through the entire journey, are there life lessons that these are things that are a definite thing that you look for in hiring people or?
Right. When you start out, I think my major problem was communications issues.
Okay.
I'll be upfront with it. Communication is one of the essential tools in building the network.
Okay, in terms of like networking with other people.
Yeah, you need to be charismatic. You need to be talking to people. If you don't talk to anybody and you want to work in your own, you'll go nowhere. And you'll be able to talk to people and listen to ideas.
Academia as well as industry.
Industry, anywhere. You just keep listening. You have 2 years, keep listening and register it here to make sure how do you process that information, and can you connect this dot, totally irrelevant dot here, to a dot here. And if you can make that connection, you see that, oh, okay, this is related straight or like this, so you know the connection. Once you map it out, then you know how to proceed from start to finish.
Okay.
Okay?
Communication is
the most essential skill anybody needs
Okay.
in success. Without communication skills, brilliant people have failed in their lives. That's very important.
Even doing pure science is hard?
Even academic sciences today is communication. Okay?
Yeah.
You have to go get grants. You need to know people. I mean, you need to understand what other people are doing. How are you different from other people? So it's a lot of communication. You got to attend conferences. You need to meet people and see what they're doing, what ideas they are pursuing, how your idea is different from their idea. Are there any connections to work together? So all these things, even in academia today, it's all communication.
Yeah. Are there other tips? Communication is the skill. Apart from communication, are there other skills that you think are very essential?
Hard work, of course. I mean,
Goes without saying.
There were days I used to spend 14 to 16 hours a night at work. That was because I had to learn. If you don't learn at that age, when you're young, you will never learn when you grow up. So, learning is one of the things that I always wanted to do, going deep. So hard work, there's no substitute for hard work.
Yeah.
Okay, and that is very critical. Communication skills is another one I adapt to it. And also focus. I learned focusing is the only way you can actually make it work for yourself. If you step on every stone that comes around you, you'll not make it. Because you have to go from here to there. You find a path, only use some steps to go there, not all these other steps in the way. Right?
Correct.
You need to focus.
Yeah. So I think communication, focus, and hard work.
Right.
If you have this,
And perseverance. You really need to, okay, I can solve it.
Yeah.
Nobody can stop me from solving it.
Okay.
And find, because sometimes you alone cannot solve it, you need equally competent people to work with. This is politically incorrect statement I will make here. In 1993, I was reading an article in Science. It was published by Dr. James Watson, the pioneer of DNA structure.
Okay.
Succeeding in science, some rules of thumb.
Okay.
And he said, I would have not played any game, even as a kid, with somebody who's not good at it. He has to be at least equally good at with
Hmm.
me. I'm winning over the guy who's not good at it doesn't make me good.
The
So
lesson
you
there.
have to win over the people that are better than you.
Hmm.
If you have that attitude, you will definitely try to win because I'm seeing you are better than me and you know how to win the game over me, but I'll keep playing with you to win over you.
Correct.
Okay?
So essentially, even choosing worthy adversaries.
Exactly. You need to have the right people because you have to grow in your career, you have to grow in your life. Play with the people that are better than you.
Hmm. To improve your own game, whatever that
Right,
game
yeah.
is.
Play with the people better than you.
Yeah.
That's one of the best advice I've read in that article.
Okay. With that thought, I would like to thank Dr. Subramanyam Vangala for taking the time out and coming here and speaking to us. We learned quite a bit about clinical trials and life lessons and some thoughts about modern, adventures, if I were to put it. Thanks for your time, sir.
Thank you.